I want to start by thanking the Lake Erie Committee for the invitation to make this presentation today. And I also want to acknowledge my co-authors who represent the various agencies listed on the bottom of this slide. This truly is an interagency effort. Understanding fish movements is a difficult thing. They're ectotherms, they live in an environment that we don't really observe directly, and they have tails. They swim around, which makes it hard to encounter them sometimes. So this puts us in a situation where if we want a deeper understanding, we've got to move beyond just basic descriptive measures of what the fish are doing to something that gives us an understanding of mechanisms and connects to the fishery management decisions. So often we will describe the patterns of movement in an acoustic telemetry network, but what we really want to understand are the mechanisms that drive those movements, the triggers and the cues. Often we can also look at the occurrences throughout a habitat and describe the conditions where these fish occur. But really what's important here is the available habitat and what habitats fish select out of what's available and how they respond to changes in that habitat availability. And then when we start to compare the habitat suitability in space and time, we want to be able to connect those habitat selection behaviors to relevant fishery management decisions. So what I'm going to do is give you 3 take-home messages right now, and then we'll get into the presentation of the results. First of all, Lake Whitefish and Lake Erie utilize only a fraction of available suitable habitats. They actively avoid low oxygen and high temperatures. I'll show you the evidence we have for that. And then finally, their behaviors in space and time make them vulnerable, more vulnerable to fisheries along the south shore than in other parts of the lake. Some of the relevant characteristics of Lake Whitefish to this presentation are that they are a benthivore, they live associated with the bottom, and they're hypoxia intolerant. They don't like low oxygen. They hang around for a long time, they're temperate and long-lived, they do migrate, and we're at the southern extent of the range. They're mainly temperate boreal species. So that means, especially in Lake Erie, that they're dependent upon summer cold water refugia. The stocks in Lake Erie are low relative to the long historic information that we have on them, and elsewhere in the Great Lakes, there are recruitment concerns. For Lake Whitefish and Lake Erie, the particular conditions that lead to hypoxia are extremely relevant because they mainly are concentrated in the central basin due to the range of depths and the position of the thermocline, which leads to a thin hypolimnion, a low volume, in which oxygen depletion can occur across a stratified season. This huge area of the central basin is potentially a very important refuge for cold water species such as Lake Whitefish and Lake Erie. And it's also the focus of the Great Lakes Water Quality Agreement, which calls for a reduction in the severity and extent of hypoxia, emphasizing this central basin of Lake Erie. We've been tagging Lake Whitefish for many years in Lake Erie cooperatively, and we've done this through a period when the network of telemetry receivers has shifted in sampling design, especially going into 2017. So the focus of my presentation is going to be these 2 years, 2017 and 2018. And this is because we have a more consistent array, but also because we have a lot of fish detected. One of the things I also want to point out from this slide is how the darker red dots, which show a greater number of unique individual fish on a receiver across the year, are concentrated along the southern shore. And so this is definitely a focus of some of the research that we're doing. Through this period, we also have the development of a hypoxia forecast model by NOAA Great Lakes Environmental Research Lab. And what it's showing us in a very prominent way is the dynamic nature of change, environmental change on the bottom in the central basin. Here you see temperature on the bottom in the top panel and changes in dissolved oxygen on the bottom in the lower panel. And one of the things we can do with this model is we can make fine-scale temporal predictions about the bottom conditions whenever a fish was detected across the network of receivers. When we make those predictions, we can look at Two things. One, we can look at the range of conditions on the bottom for all of the receivers across the Central Basin, which is what you see in the gray circles that are all overlapping on these plots for each year. The other thing we can do is we can plot an overlay of the conditions on the bottom where whitefish were detected, and that's shown by the blue dots. The yellow lines demarcate what we expect from the literature to be the oxythermal niche for lake whitefish. And by and large, these fish generally remain in cooler waters as we predict, but they also were detected in areas where the bottom conditions had low oxygen, outside of what we would expect to be their oxythermal niche. Another way to look at these same data is to look at the time series through the stratified period when we were able to make predictions from the hypoxia model. And in each of these years, you can see changes in the available habitat. This is the number of receivers, or the proportion of receivers, in which we had suitable oxythermal conditions predicted for lake whitefish. This changes dramatically and even approaches zero in 2017. And here you see very few detections during this period, as we might predict. But the big thing here, the big take-home message, is that they're really only utilizing a very small subset of the available habitat as indicated by the receiver network. Up to now, I have demonstrated to you just a static empirical description of what data we have so far. But what we know is that these fish are moving around, and when we start to look at the dynamic nature of these movements and pair up where fish were detected between two receivers, we can start to ask some more interesting questions with the model predictions. And here what you see is paired observations where we look at the conditions on the bottom where the fish started in one time step and where they ended up as a function of the change, in this example, in dissolved oxygen between those two detections. And one of the things that's really interesting is that we can see when the dissolved oxygen on the bottom was low where the fish started, they often moved to an area that had higher dissolved oxygen. In other words, in the positive direction for dissolved oxygen. We can look at these paired observations in a different way and also ask questions about the change in temperature. And here in this plot, you can see that when the temperature was basically on the warmer end of what was observed, these fish tended to move toward a cooler temperature, sometimes by several degrees. And this is something we might expect, but it does emphasize, as well as the information on hypoxia, that these fish are actively moving towards cooler temperatures or moving away from low oxygen conditions. This is certainly a work in progress, and there is some uncertainty in the model predictions relevant to what the fish are doing in Lake Erie because we don't know at what depth these fish were when they were detected. So these fish may very well be above the poor quality habitat conditions that we observed, but they still may be responding to them in some way for various reasons. One of the things we were able to do to help investigate this further was to deploy acoustic data storage tags in Lake Whitefish in the fall of 2019. And these tags not only transmit temperature and depth, but they also record temperature and depth. And as a little teaser, Andy Cook was able to recover from the fishery 3 of these tags so that we could look at the continuous record of information. And that's what you see in this plot. And one of the things we will be able to do this coming year is match these data up with detections across the network. So what you see is more through June through August, more narrow range of depths occupied, a wider range of temperatures, but still cooler temperatures on these tags. And then a shift in September and October that represents deeper habitats as well as shallower habitats late in October, presumably after turnover and associated with a westward movement of these fish back to the western basin. The other thing that we've done is to deploy data loggers, at least in US waters in 2020, on the acoustic telemetry network. And this animated GIF here shows you at least a first look at what kind of information we're getting, where we can match up, again, some of the conditions on the bottom with what these fish are doing, and potentially also improve the hypoxia forecast model. So I think you can appreciate that we have a lot going on with Lake Whitefish telemetry in Lake Erie, and we're trying to get at some fairly complicated questions. So to sum things up and to emphasize what those questions are and hopefully link back to those key take-home points, I want to point out that the data is showing and continue to show each year that the movements of the fish we've tagged over in the West Basin are concentrated along the southern shore. There may be something stock dependent going on there because we don't know if we have representatives from, for example, Long Point Bay, or if there are still individuals out there from some of the historic concentrations of Lake Whitefish and other parts of the lake, like the northern part of the Central Basin. Another sort of pervasive question here is that if we are successful at achieving some of the Great Lakes Water Quality Agreement goals, will this change patterns of behavior and habitat use? So I think that continuing our investigations to understand things mechanistically is one way in which we can try to answer that question that is relevant to fishery management. Finally here, how does habitat suitability influence the fish behavior in a way that influences the fishery interactions? So as hypoxia develops and moves around, I think we need a better understanding of how this changes the vulnerability of these fish to the fisheries, to the different fisheries across the lake. And one of the other things that I'll point out that is forthcoming is that we have other species tagged in the lake that utilize these habitats but still respond to hypoxia and changes in habitat suitability in ways that are relevant for comparisons with Lake Whitefish, and in a way that may help us better understand mechanistically what's going on. And I'll stop there and hopefully have time for questions. Thank you.